Now that we've covered DNA replication, we are going to take a look at some of the techniques that have been utilized to sequence DNA and manipulate it as well. So we're going to start by looking at DNA sequencing experiments. These are conducted in vitro or in a test tube or a little microfuge tube. And it uses the same components that are required to replicate DNA in a living cell. The dideoxy sequencing method was developed by Dr. Frederick Sanger. He was also the first to sequence a protein and then again DNA. He received 2 Nobel Prizes for his work in sequencing the protein and DNA. It is pretty rare to have accomplished the awards of 2 Nobel Prizes. So we are going to first cover the components that are required in the reaction mixtures. We need a template DNA strand. So this is the DNA of interest. We need a small primer. So this is a small short sequence that will find a complementary sequence in the template strand. We also need the 4 DNTPs, ATP, GTP, TTP, and CTP. And these are radioactively labeled, often with P32. We also need 1 dideoxy NTP, and we'll talk about how this ratio varies. And of course, we need a DNA polymerase. And what we do is make 4 reaction mixtures, each with a different dideoxy NTP. Elongation of the primer strand, so we're going to be copying that template strand, stops when the dideoxynucleotide is incorporated into that growing strand. So that means that DNA elongation will terminate at different positions along that growing strand, and that will enable us to sequence that. We'll talk about how that's done. So this slide shows what happens when a dideoxy ATP is added to a reaction system. So here's our 2,3 dideoxy ATP. So lacking that 3' hydroxyl, it is unable to accept a new nucleotide, which is required. So here's our template sequence. Remember, DNA is copied 5' to 3', so we've got a 5' primer. We have a reaction mixture that contains our regular DNTPs and then that DATP. And this is DNA polymerase 1 utilized in this reaction. So as we can see, this particular strand ended at this point, whereas this one got a little bit longer. So what we end up having are many different lengths of the DNA replicated, and that will allow us to line them up and sequence this to completion. So the DATP mixture is about 98% total A trinucleotide and about 1 to 2% of the dideoxy ATP. So most of the time, a dideoxy ATP will be inserted in the sequence and replication will continue past that A position. However, 1 to 2% of the new DNA chains will be terminated at each point where a dideoxy ATP is inserted and replication stops. So this experiment is then repeated with other dideoxy NTPs, giving the same type of fragments. that will terminate at whatever that dideoxy NTP is. So, having created a collection of 4 different dideoxy DNA termination mixtures, the next challenge is to separate and identify the places where chain termination occurs. And this can be accomplished using electrophoresis on a polyacrylamide gel, or another more specific is PAGE, P-A-G-E, polyacrylamide gel electrophoresis. And basically it involves the net migration of DNA fragments that is proportional to the size of the fragment and its negative charge due to the phosphate residues. 4 mixtures are loaded on the gel side by side in sample wells, and the electrophoresis is carried out. The DNA strands synthesized using P32 radiolabeled they were synthesized using those radiolabeled DNTPs, so these bands can be visualized using X-ray film, and we'll take a look at that in a moment. And then the DNA sequence can be directly read from the binding pattern, or the banding pattern, in the 4 lanes on that X-ray film. And this is an example of what one of those sequencing gels looks like. This sequenced about 200 bases. And here's our G lane, A, T, C, and you just read across left to right, 5' to 3'. Here's our A, here's our T, here's our G, C, C. Depending upon the resolution of these gels, this one is pretty easy to read, but some are not as clear as this. But what you can see, we get this segment, and then we have an overlap here, and we have an overlap here, and so forth. So in order to continue sequencing whatever this DNA is, we must synthesize a new primer that corresponds to a short DNA sequence near the 3' end of this sequence, and repeat the experiment. So now we're going to talk about another technique that is utilized, and that is polymerase chain reaction, or PCR. In many cases, the DNA material available is in very tiny amounts. 2 examples of this is in forensic criminal investigations or in biological samples involving animal hair or scat samples. So many of the methods that we've discussed, or others we haven't, for characterizing DNA, including the sequencing methods we just looked at, will not work with such small amounts of material. So before these methods can be used, the small DNA samples must be amplified to give larger amounts of material. And PCR is a method for amplifying small amounts of DNA. It was developed about 20 years ago by Kary Mullis. He received, Nobel Prize was received for this work in 1993. And Mullis has written that he conceived this idea while cruising the Pacific Coast Highway one night in his car. I don't know if that's true or not, but PCR is basically a DNA synthesis system like we have been looking at, except with one important modification. And that is the use of Taq polymerase. So the objective, amplify a small amount of DNA. So we need, as far as the reaction components are concerned, the DNA sample. We need oligonucleotide primers to define a region, 40 DNTP DNTPs, and Taq polymerase. This is a heat-stable DNA polymerase, and this is the very important modification. So this polymerase, because of its heat stability, is able to undergo repeated cycles of heating and cooling. And this is isolated from a thermophilic bacteria. So this is the protocol or the cycling that occurs in the PCR machine. Of course, we've already assembled the reaction mixtures. We incubate that in the thermocycler for 20 to 30 cycles. So that means we're going to heat it, cool it 20 to 30 times. At the high temperature, the DNA strands separate. And as the temperature lowers, the primers anneal and DNA synthesis can occur. So this slide depicts PCR products, 3 rounds of PCR products. So in round 1, we have 2 strands. We have the DNA template, and they are separated into 2 strands when that mixture is heated. As that cools, our primers anneal and allow DNA synthesis to occur. So at the end of one cycle and the beginning of the next, we will now have 4 templates that undergoes the same cycling process. We heat it, strands separate. As we cool, the primers anneal and replication occurs. And this is repeated again 20 to 30 times depending upon the length of that DNA. So after 30 rounds, we've got about 30 run-on copies of millions of short DNA sequences of interest. And then that DNA can be sequenced and characterized by the other methods we have discussed. Now we're going to take a look at how we can manipulate DNA, and that is by making recombinant DNA. And what that means is we're going to take a DNA of interest. Typically this is going to be a gene, although it could be another section of DNA that we want to characterize or experiment with. But we need a DNA source. We need what's called a cloning vector. So the cloning vector is, what do I want to say here? It is a mode of transferring that DNA into another cell so it can be replicated and characterized. We need restriction endonucleases. So these are enzymes that will make specific cuts in the DNA. We also need a DNA ligase, and that does exactly what it says it does. We've seen ligases before. And that will covalently ligate the DNA segment into that cloning vector. And then we need a host organism. So this is some sort of cell, or it could be yeast, depends upon what kind of work you're doing. But it is a host in which the plasmid can be grown and amplify the DNA of interest. And these cloning vectors can be plasmids or viruses, depending. So the process of cutting and pasting segments of DNA basically is the bread and butter of molecular biologists. The cloning of DNA then involves, as I said, restriction endonucleases that cut at specific restriction sites in sequences. And the restriction sites in double-stranded DNA are often described as palindromes, although really they're kind of an inverted cousin of a palindrome. But a palindrome is a series of letters that forms a meaningful word or series of words that can be read identically in the forward or backward direction. And here are 3 examples of palindromes. Some simpler than others. Noon, read the same, forward and backward. Madam, I'm Adam. And here we have Madam, I'm Adam. And a man, a plan, a canal, Panama. Reads the same way in the reverse direction. But let's look at what a real endonuclease restriction site looks like. And here is the EcoR1 restriction site in double-stranded DNA. And I just want to note that each restriction enzyme has its own unique restriction site or sequence. And there's probably, well, I know there is a table of some of these in your text. I also want to point out that, and we'll take another look at another slide momentarily, but some enzymes produce blunt ends and are more difficult to work with. However, the EcoR1 site produces what we call sticky ends, and that makes life easier. Okay. So here is our inverted palindrome. This GAA is read GAA from this side, and our CTT is replicated up here. The cleavage site is right here and right here. And what that means is that on the lower strand, the G goes with the other half here, and the C is an overhang. So there's nothing connecting it to the bottom strand anymore. And the same applies up here. While we have the A in the upper strand, the T is missing in the lower strand. So these are our, whoops, wait a minute. Yeah. Did I say that right? G is not here, C is here. So we do have overhangs. Might have screwed that up a little bit. Anyway, so the eco refers to E. coli, and it is, this EcoR1 was the first enzyme that was isolated. The R1 tells us it was the first such endonuclease isolated from that organism. Most restriction sites contain a palindrome, but only of the sequence of bases is read halfway on the strand, as I indicated, and half on the other strand. I want to see if there's anything else I wanted. Oh, if we cut both of our plasmid and our DNA with the same EcoR1 enzyme, then they both contain sticky ends, which helps them anneal better compared to those blunt-ended. However, there is an issue with this. Because of those sticky ends, that cloning vector can religate on itself, and we'll discuss a method to avoid that possibility. So here's a slide that shows us some of the different different restriction sites. And I did. Let me go back one sec. Can I go back? No, I guess I can't. Here's the EcoR1 we just looked at. Here's BAMH1. Again, we have, you can see that inversion here of that palindrome. So these are all enzymes that create sticky ends, as is HIN3. However, these two create blunt ends. So there are no overhangs here. And it takes a little extra work to get a cloning vector with DNA in it that contains blunt ends, but sometimes you don't have any other choice. So what I want to discuss is directional cloning. And in order to do that, so what we want to do is, number one, if we cut the vector and the DNA with the same enzyme, a number of things can happen. One, the vector could close up on itself with no DNA in it. We could get a vector with the DNA in the correct orientation, or the DNA in the incorrect orientation. So 3 possibilities. And we can use a method called directional cloning to eliminate those chances of getting the other 2 products that we don't want, essentially. And that's directional cloning. And what is involved there is you cut the vector and the insert with 2 different enzymes. So the insert can go in only one way, and the vector cannot religate on itself. Okay. Now we're going to take a look at the basic procedure, and that will conclude this segment of DNA sequencing and some techniques that can be applied to DNA. So the basic procedure here, and this is just using an EcoR1 restriction site. So we have these 3 segments here. We have EcoR1 sites. We get this, when we cut this with the EcoR1, we get 4 different fragments. And this is just an application where we're not actually inserting a specific gene of interest, but we are rearranging the order of these sequences and seeing what the effect will have on function here. So when we re-anneal these, we can get a different order and obviously numerous products. So sorry, lost my train of thought. So this allows for hybrid products then, which we can characterize and study with respect to function.